An experimental drug called NU9, discovered here at Northwestern University and approved for clinical trials for the treatment of ALS, also improves neuron health in animal models of Alzheimer's disease, according to a new Northwestern study published in PNAS. This discovery is giving scientists hope that the drug could be effective in multiple neurodegenerative diseases by addressing the underlying mechanisms of disease. Today, I welcome the study's co-corresponding authors, Richard Silverman and William Klein, to talk about this discovery. Richard Silverman is the Patrick G. Ryan Aon Professor of Chemistry and Professor of Pharmacology and invented NU9. Dr. Silverman previously invented the blockbuster drug Lyrica to treat nerve pain and epilepsy. William Klein is a professor of neurobiology and neurology at Northwestern and an expert in Alzheimer's disease, and leads a pharmaceutical company that has a therapeutic monoclonal antibody to treat Alzheimer's disease currently in clinical trials. Welcome to the show.
Thank you.
Thank you.
It's so nice to have you both here to talk about some really exciting developments in this drug, NU9, that Dr. Silverman, you invented here at Northwestern University. Can you take me back to the discovery of this molecule and the path to developing it and how it works?
We're talking, 15 years ago, actually. And another professor here at Northwestern, Rick Morimoto, came by and said he was interested in protein folding. He was starting a collaboration with somebody in a small company who was interested in his assay for protein aggregation and wondered if I would be interested in doing medicinal chemistry for this project to try and find some molecule that would be important to inhibit proteins that aggregate and cause neurodegenerative diseases. And the assay that he had specifically worked on was related to the disease ALS, amyotrophic lateral sclerosis. And so that's what we started on. And we had what's called a high throughput screen. This is many compounds. It was 50,000 compounds that were screened in the Morimoto assay that had been converted to this high throughput format. And they looked for molecules that inhibited protein aggregation from a mutation that's known to cause ALS. And we got quite a number of molecules that did that. In fact, too many. And so it was part of my job and other computational chemists to sort of hone down these numbers of compounds into something that was more manageable to modify structures and make something useful. And so between the two of us, then, we're able to hone it down to 3 separate classes of structures that had multiple molecules in those classes that were active. And so we knew it wasn't just, a one-shot pony. It really was something that was reproducible. And so that was my job. I had these 3 classes, and I was then assigned make them more potent, make them better.
If you're enjoying this episode of Breakthroughs, please share it with a friend. Now back to the show.
And so over a period of 8 years, something like that, I had students and postdocs in my group modify the structure. We couldn't do it by classical medicinal chemistry approaches where you know what the molecule's binding to that's causing this effect. We didn't know what the target protein was because we discovered this through what this is called a phenotypic screen, where we're not looking for something to block a particular protein, but we're looking for molecules that prevented proteins from aggregating, and we didn't know why. We just wanted to inhibit the proteins from aggregating so that we could prevent the ALS. And so typically, when you know what the target is, you can then get crystal structures, you can design molecules that have the right shape, the right structure, and build from that. So we were going blind. Basically, I knew the structures that were active, and now we just had to do random modifications at first to see where on the molecule can you change it and make it better. And so then once we saw what parts of the molecule we could change, we then started to put other groups on. And that's why it took so long to do that. And so then we ended up with 3 molecules that we felt were optimized and would be ready to move forward. And that's when we started to do pharmacokinetic studies to see, are they metabolized rapidly? Are they not soluble? Can they cross the blood-brain barrier? Because this is a disease in the brain. The molecule has to get into the brain. And so that was many more years to hone the structures to something that then had the properties that will allow you to take a pill and then get it to where it needs to get, which is the neurons in the brain. And so we optimized those 3 classes. And of the 3 classes, one seemed to be superior to the other 2. And that molecule then is this molecule NU9 that is the basis for that PNAS paper you talked about.
NU9, just tell me about the name.
Coming from Northwestern, I thought I'd been here now 49 years, so I bleed purple. But the 9, strangely enough, in the paper that we published that had that molecule in it, it was the 9th molecule in the list. And so I figured, all right, we'll go with that.
Lucky number 9.
Yes.
And this really ended up being more than potential. You were able to actually test this in animal models, and now it's been approved for FDA clinical trials for the treatment of ALS. Tell me where all this stands right now with that project.
Yeah, so it's very expensive to do studies that the FDA wants to approve for clinical trials, and then even more expensive to do the clinical trials. And so I started a company and licensed NU9 from Northwestern into my company. The company's called Akava Therapeutics. And so the compound then changed names when it went into the company. It's called in the company AKV9. I kept the 9 in that one too. And so we raised funds to do all the studies the FDA wants to decide if it can go into humans. And that took a couple of years to do that. And then we filed all the paperwork with the FDA, and we had done lots of studies in animals for efficacy, whether it works or not, and also whether it has the right pharmacokinetics, whether it can get to where it needs to be, and also toxicology, because it has to be a safe drug. And we found that you can use really large doses of this in the animals, and it doesn't hurt them. That whole document was sent in and then approved.
So we haven't started the clinical trials yet, but this is all your ducks are in a row at this point.
Yes, all we need is the money
right.
to do that, because now it's in my company and it's up to my company to raise the funds for the clinical trials.
Well, there has been a lot of excitement about the potential of this drug. And Dr. Klein, I want to bring you in because you're an Alzheimer's disease expert. And ALS and Alzheimer's disease do share some important cellular and molecular mechanisms. Can you tell me about those similarities and what made you think that NU9 might work for Alzheimer's disease too?
Well, Rick made me think that. Ordinarily, scientists are terribly skeptical. And, we're taught, don't believe anything that you're not forced to believe. And Rick alluded to the fact that NU9 can affect several different protein compounds that give rise to ALS. And to me, that was the real clue for going with his suggestion, let's try it for Alzheimer's too. So we had discovered quite some time ago a novel toxin that now most people think is responsible for the onset and progression of Alzheimer's disease. It's a normal metabolite that sticks to itself and is kind of like a hormone from the dark side. It's different from amyloid plaques that everybody knows about, but it's evolving into the actual nasty culprit that's involved. So Rick suggested, why don't we test NU9 to see if it blocks the formation of this compound called an amyloid beta oligomer. It's about the size of a normal protein.
And that's an aggregate also, which made the connection.
Exactly. So there's a PhD student in chemistry that worked between our two labs to collaborate in doing the cell biology and toxicology in my lab on Alzheimer's with the compound that Rick and his team had developed. And lo and behold, it was very exciting. If you subject these nerve cells, which we can grow in a dish, to the precursor, the stuff that does aggregate, normally the neurons will take it out of the medium, and inside their cells, they will make the toxins, because that's the tendency of this protein to self-aggregate. And we have an assay to measure it. Interestingly, the way we measure it is with an antibody that also is named NU. So
That's really confusing.
we came from different directions and ended up with the same names. We have NU1 through NU20. It turned out to be remarkably successful in these initial experiments. And we found that the mechanism, which we're still chasing down for its absolutely precise molecular target, was not exactly what we might have expected. It didn't block the formation of these aggregates, but it accelerated the removal of them. So there's this process that's going on within the neurons that gets rid of garbage, in essence. And these toxins, these toxin precursors, when they self-associate, really slow down that process, and that's why they tend to accumulate. But NU9 accelerates the basis for clearing it out. The garbage truck coming by 3 times a week instead of just 1, and the trash just never builds up in the cell, and you've got some rescue going on. And then we go from there to animal models. And naturally, you start in a test tube, then you work with cells, then you go to animal models. And we have animal models that develop pathology very much like Alzheimer's disease. And lo and behold, the NU9 was terrific at blocking the accumulation of A-beta aggregates, or these oligomers, in the brain. And they block the form of Alzheimer's pathology that is induced. So it makes the toxin not accumulate. The effects of the toxin are no longer there. So it's a successful compound in our trials.
This was the PNAS paper. This was a small proof of concept study, as you just described. But have you ever seen anything like this before in your work with Alzheimer's disease?
What I think is that this is a very exciting molecule. And as we study more about the mechanism, we can begin to see that it makes sense. There aren't compounds like it, but there's thoughts about the basic mechanisms that make sense in terms of what people had already established.
So what happens next? You're already on this road to clinical trials with ALS. So now for Alzheimer's disease, what's the path forward here?
The first phase of clinical trials is with healthy subjects. And so you just want to find out at what dose do you start seeing some side effects. And so you do a study with healthy subjects where you keep increasing the dose until they start complaining about something. And then that's as high as you ever want to go. So that doesn't matter what the disease is, because safety is safety, and that's what it is. So then it splits off to phase 2 with patients, and you would have to then do 2 separate clinical trials, one with ALS patients and one with Alzheimer's patients, and then see what dose you need for each of those diseases, and then you'd have the trial go for as long as you can. And something like ALS, unfortunately, these patients die very quickly. I think it's 90% are dead within 10 years, 50% within 5 years. And so you can't have a normal, long clinical trial. Your clinical trial is typically about 6 months, because many of these patients will be dead in a year or 2. And so you have to have a drug that's effective enough that you could see a difference between the placebo and the treated in a 6-month period. And so those are short trials. There also are not that many people with ALS, although you hear about it all the time. It's what's known as an orphan disease. There are less than 200,000 people at any given time in the U.S. with that disease, partly because they die so quickly, so they don't accumulate. A disease like Alzheimer's, however, there are millions of people that have that. And so you can run trials with many more people. And of course, the FDA then wants you to run trials with many people, and you can run it longer because you may not really see an effect in 6 months. You may need to dose for a couple of years before you really start seeing differences. And so that's a much more expensive clinical trial, more people and longer time. And so you have to separate those two.
Everything that Rick said is spot on. And with respect to the Alzheimer's trials, we're still in some serious preclinical phase research. And that's not in the cost of hundreds of millions of dollars to do that. But, given the current situation in federal funding, even something that's as promising as NU9 for preclinical research for Alzheimer's disease is kind of stuck in the weeds in Washington, D.C.
What do you want the public to know about the potential of this drug? Why should people maybe be invested in seeing this move forward?
Yeah, I think as we've seen in ALS, it's effective against mutations that are completely separate from one another. They're not related to each other. They work separately, and now they work in Alzheimer's models. We want to then try it on other neurodegenerative diseases. One of the things it does is improve the health of mitochondria. And so there are a lot of mitochondrial dysfunction diseases. And so this could spread out into a lot of different neurodegenerative problems. And so this should be exciting, I think, to more than just the relatively few ALS patients. This is something that I think could be useful for many different diseases.
And the compound itself is close to the bullseye in the common mechanism that unifies so many of these degenerative diseases. So the fact that ALS proteins, they're good proteins, but they've gone bad. In Alzheimer's disease, you have good proteins that go bad. This is a drug that can affect both, can clear them both out when they go bad.
Well, I would say that this could be a game changer. Usually it's, one drug per disease, and you change diseases and you have to change drugs because the scientists are focused on a single target that is important for that one disease. And then when you go to another one, that target may not be so relevant. So you have to find another molecule for a different disease. Whereas this one is hitting whatever the target is, but it's hitting pathways and mechanisms that are common to all of these diseases. And so this really could affect multiple diseases with the same treatment.
So from my view, the more we learn about NU9 here, the more excited I get about it. And I think that kind of excitement is legitimate.
Well, thank you so much, Dr. Silverman and Dr. Klein, for being on the podcast, sharing this remarkable story. We can't wait to see what happens next with NU9.
Thank you.
Thanks for the invitation.
Thanks for listening. Please click the bell to receive notifications about our latest episodes and follow us on social media at NU Feinberg Med to stay up to date with our latest research findings.